Have you ever thought about how your body copies its genetic code billions of times? And what happens if it slips up just one letter? One typo may look like a small error in chats, but one wrong letter in your DNA can change everything. DNA replication is a process by which the body copies over 3 billion base pairs during cell division, maintaining accuracy in passing the genetic code to each new cell. But, even in this highly accurate system, small mistakes like missing or extra bases can still occur. When replication mistakes go uncorrected, they can turn into permanent mutations that affect how cells function, leading to genetic disorders or cancer. DNA replication is precisely regulated system as well as an almost perfect phenomenon through which an exact duplicate of the genetic materials becomes available for each new cell. The process begins with helicase and twisting the double helix at the replication fork, where the two DNA strands are separated. Then, the new complementary nucleotides are added, adenine with thymine and cytosine with guanine, so that a new strand is formed that is a mirror image of original one. DNA polymerase also proofreads the process, so errors on the way of synthesis can be detected and corrected. All these steps are focused on preventing mistakes and maintaining genome completeness, because without them, mutation could keep occurring. In perfect order, the steps could be uncoiling strands, matching bases, and proofing the build confidence that genetic information is copied accurately. Through its a carefully regulated process, DNA replication can still make errors from time to time, requiring the cell to make use of repair mechanisms to maintain genetic stability. This means that even though the process is careful, mistakes can still happen. To fix this, cells have special ways to find and correct errors. Cells use systems to check the DNA and repair any problems with the base pairs. Even though enzymes usually catch and fix errors during DNA replication, some mistakes manage to escape detection. When these are not fixed, they get copied during cell division.
Prey explains that once this happens, the cell sees the error as normal and it stays in the DNA as a permanent mutation. Although these repair systems catch most mistakes, small uncorrected errors may become permanent mutations, leading to genetic issues. Cells have repair systems with enzymes that team up to find and fix DNA problems. Keeping the genetic code stable, operation mechanisms include cut and replace, which explains the types of damage and repairs the DNA structure. For instance, in base incision repair, a damaged base such as a uracil by a DNA glycosylase, thus creating an apurinic or apurimidinic site that would later be cleaved by AP endonuclease, followed by correct nucleotide by a DNA polymerase, and sealing of the strand through action by DNA ligase. Furthermore, mismatch repair, in which the enzyme completes, finds, and excises mispaired bases, results from replication and uses the original strand as a template to repair the sequence. NER, like mismatch repair, consists of a bunch of endonucleases making two or three cuts to surround a DNA segment around the large lesion, to be replaced and sealed by a polymerase and ligase subsequently. To summarize, these enzyme-based repair systems such as NER and MMR play as an indispensable role in the maintenance of genome stability and the prevention of permanent mutations. Though rare, these uncorrected errors can lead to permanent mutations that disrupts normal bodily functions, impair protein activity, and eventually cause severe inherited genetic disorders, such as sickle cell disease, which results from a single mutation in the beta globin gene. This mutation shapes the red blood cells in a way that can obstruct blood flow and consequently produce painful complications. Another case is cystic fibrosis due to the insertion of an extra DNA sequence in the CFTR gene. It leads to the expression of a defective protein that obstructs the lungs and digestive system, tract with thick and sticky mucus. In the case of Muir-Gorlin syndrome, mutations in genes responsible for DNA replication resulted in poor cell growth and divisions that culminated in a short stature and underdeveloped bones. These mutations highlight different types of replication errors that would have caused serious permanent genetic disorders if left uncorrected. In some cases, when the mismatch repair system fails to fix errors missed by DNA polymerase, mutations build up quickly, increasing the risk of cancer. The loss or silencing of important MMR genes such as MLH1, MSH2, and MSH6 can cause Lynch syndrome, a condition linked to a high chance of developing colorectal and other cancers due to microsatellite instability. MMR failure also leads to chromosomal instability, which causes abnormal chromosomes, division and changes like deletions or duplications that supports cancer growth. In addition, loss of heterozygosity weakens tumor suppressor genes, which disrupts normal control of cell division. Overall, these problems increases the risk of cancer and make treatment more difficult. Errors in DNA replication can contribute to aging, genetic variety, and the process of organisms over the time. The act of DNA replication transfers genes at each cell division. When all is well, it is the cable that transfers a fateful message to the offsprings from the parents. Changes in the genome during the DNA replication. The changes occur during the DNA replication when organisms are supposed to rehearse and pass on the changes from one generation to another. When things go wrong, replication can, because this cycle becomes slow as one ages, more replication errors starts to pile up, while the shortening telomere the chromosomes initiates pathways that drive cellular aging and death. Thus, DNA replication aids inheritance and progress, whereas it is a factor in the aging of an organism. If a small error in the replication is not corrected in the next generation, it can become a big thing. Therefore, DNA replication can also create an avenue for the determining fate of a species. In conclusion, although DNA replication is a very accurate, errors can still occur. The cell have repair mechanisms to correct most errors, but some slips through, resulting in permanent mutations. These mutations can cause genetic diseases, contribute to aging, disrupt cellular function, and even results in cancer. The impact of this even in a minor error can be significant and long-lasting. In addition, ongoing research into the DNA errors and repair mechanisms offers hope for preventing and treating genetic disorders, ultimately improving human health.